Good morning everyone. I hope you all in good health and high spirit. Welcome to today's lecture on topic electromyography, abbreviated as EMG, and electroencephalography, abbreviated as EEG, that lies at the intersection of human physiology and engineering. Before we dive into the intricacies of EMG and EEG, I want to take a moment to express my sincere gratitude to each and every one of you for joining us today. Today, I have the pleasure of guiding you through the captivating world of EMG and EEG, a technique that allows us to peek into the intricate electric activities of the muscles and brain and gain insights that were one thought to be impossible. Whether you are a seasoned student in the field of biomedical engineering or someone who is new to the concept of EMG and EEG, I assure you that this session holds valuable information and perspectives for all. From the fundamental principles to applications, we will be covering a wide spectrum of knowledge today. So, without further ado, let's dive into the realm of electromyography and electroencephalography. From the basics to the nuances, we will cover the mysteries of muscles action potential to brain wave and explore how this technology is changing the way we perceive the human health. Once again, a warm welcome to each of you. Thank you for being here, and let's make this session enlightening experience. Let's get started. In our previous session, we have already covered the basics of electromyography and discussed in detail the apparatus required for capturing the electromyography. In today's session, we are going to continue with electromyography. In this, we will be covering the topics motor unit structure, characteristics of EMG signal, types of EMG, and applications of EMG. In electroencephalography, we will covering what's an electroencephalography EEG is, highlighting the importance of EEG in understanding brain functions and diagnosis neurological disorders. Then we discuss the fundamentals of electroencephalography and the applications of electroencephalographies. Now, let us move on. What is the structure of a muscle? EMG is a technique for evaluating and recording the electrical activities produced by the skeletal muscle. The electrical signal is results of the muscles being activated by the nervous system. The greater the stimulation, either by high frequency firing of motor neurons or greater recruitment of motor unit, the larger the electrical signal will be. Zooming in, the motor neurons and muscles join each other at the neuromuscular junction, which is also known as motor end plate. This is a specialized synapse where the motor neuron junction with each muscle's fiber. The region comprising all the neuromuscular junction is called the innervation zone. From this innervation zone, the action potential propagates to either sides of the muscles. If we zoom in at microscopic level, a motor unit comprises motor nerves and the muscles fiber, that is the intravased. In figure A, it illustrates the motor unit structure including the motor neurons, muscular fiber and motor end plate. The structure of the motor unit is visualized in figure A. It is the smallest functional unit of a muscles. Each muscle has multiple motor unit. Motor units are recruited in a particular order. referred to as recruitment order. To generate a small amount of force, the nervous system recruits only small motor unit. As the required muscles force increases, the nervous systems recruit progressively large motor unit as well. Now, a basic motor unit structures include the motor neurons, motor muscular fibers and motor end plate. Motor unit consisting of muscular fibers, they are different types of fibers, but within a given motor unit, all fibers are of same time. Based on the contractile speed, muscles fiber can be either fast twist or slow twist. Muscles fibers are not only distinguished by their speed of contraction, but also by their fatigue characteristic. Slow twist fiber tend to resistance to fatigues, while the fast twist fiber fatigues quickly. Generally, fibers are categorized as type 1 slow fatigue resistance, type 2A fast moderately fatigable and type 2B very fast highly fatigable. Now, let us discuss some important characteristics of EMG signal. EMG measures the sum of the electrical activities of multiple motor unit. These are the motor unit action potential, abbreviated as MUAPs. This is observed as bipolar signal with a symmetric distribution of positive and negative amplitude. Mean values equal to 0. Raw EMG spikes are of random shape. The observed signal is influenced by the recruitment of the motor unit and their firing frequency, as shown in figure B. In figure B, you can visualize the influence of motor unit firing and recruitment on the superposed surface EMG signal. The amplitude range of an EMG signal is 0 to 10 millivolt. Before amplification, the amplitude has the potential to provide a measure of the magnitude of muscles forces, but this relationship is complicated by both the characteristic of the measured EMG and muscles mechanics. Typically, the frequency of an EMG signals range between 6 to 500 Hertz, showing the most frequency power between 20 to 250 Hertz. An EMG signal is influenced by the firing rate of motor unit, which in most condition fire in frequency range of 0 to 20 Hertz if multiple motor unit firing is involved. Now, let discuss an important terminology associated with the EMG. It is the maximum voluntary contraction, MVC. A measure often used in EMG is the maximum voluntary contraction, MVC. The MVC is the maximal force generating capacity of a muscles or group of muscles. The MVC is used to measure and evaluate the muscle strength and provide physiological references. The MVC is often used to normalize EMG signal, and other muscle contractions are then expressed in the percentage of the MVC. This make comparison of the signal pattern within and between study particularly possible. Now, let us focus on the different types of EMG. There are several types of EMG. The most important one are surface EMG, abbreviated as SEMG, high density EMG, abbreviated as HDEMG, and needle EMG. They differ in the placement of electrode and number of electrode used, and therefore, in the data obtained. One of their difference is the scope of the technique. Figure C illustrate the scope of various EMG techniques. Surface EMG, it is a non-invasive technique to measure the muscles activity, where the surface electrodes are placed on the skin overlaying a muscles or a group of muscles. In clinical neurophysiology, the surface electrode is widely used to record compound motor unit action potential, abbreviated as MUAPs. Beside this clinical use, SEMG is used to record on-off switching of muscles and to estimate muscles force during voluntary dynamic contraction. SEMGs consisting of single bipolar muscles form two electrodes placed on muscles. These techniques do not allow for single motor unit information and proper data quantification is needed, especially when the simultaneous recording from multiple electrodes need to be interpreted. Now, let us talk about the high density EMG, abbreviated as HDEMC. It is a non-invasive technique to measure electrical muscles activity with multiple more than two closely but evenly spaced electrodes overlaying a restricted area of the skin. High density EMGs, HDEMG, is a non-invasive technique to measure electrical muscles activity with multiple more than two closely but evenly spaced electrode overlaying a restricted area of the skin. The configuration of the electrode can be either in a linear array or in a two-dimensional matrix. Beside temporal activities, HDEMGs also allows spatial EMG activities to be recorded, thereby expanding the possibilities to detect new muscles characteristics, especially the evaluation of single motor unit characteristics. The last category of EMG is the needle EMG. For needle EMG, the electrodes are inserted into the muscles. The electrode are mostly used to evaluate motor unit function. Single motor unit information can be recorded rather easily with needle EMGs because the distance between the active muscles fiber and the needle electrode is small. As needle EMG records signal invasively, the signal characteristic and thus the amplifier requirement differ quite significantly from the regular surface EMG measurement. Needle EMG only record activities from a small area of the muscles. So, these are all about the various types of EMG. Now, the applications of EMG are: EMG can be used in several fields. As mentioned previously, needle EMG is used in the clinical, neurophysiological and diagnostic tool. HDEMG has been more research focused. Beside clinical use, EMG is used in rehabilitation, research and sports science. So, in summary, EMG is a tool for evaluating and recording the electrical activities produced by the skeletal muscle. It is the sum of the electrical activities of multi-motor unit, and EMG signal is bipolar signal with a symmetrical distribution of positive and negative amplitudes. There are several types of EMG, includes surface, high density and needle EMGs. With this, we completed our first part of today's session, that was on electromyography, EMG. Now, it is time to dive into another interesting topic, that is the electroencephalography, most commonly abbreviated as EEG. So, first of all, let us find out what electroencephalography EEG is. It is a non-invasive neurophysiological technique used to record the electrical activities of the brain. This technique involves placing a set of electrodes on the scalp, which detect and measure the electrical signals produced by the neurons, brain cells, as they communicate with each other. The brain electrical activity is a result of collective firing of neurons, and these electrical signals can be categorized into different frequency bands such as alpha, beta, delta, theta and gamma waves. Each of these waves type is associated with a specific state of consciousness such as relaxation, alertness, deep sleep and more. EEG is valuable for studying brain functions and understanding various aspects of cognitions, emotions and neurological disorder. It is commonly used in both clinical and research setting. EEG recording provides a real-time information about brain activity and can help identifying abnormalities, diagnose conditions like epilepsy, monitor changes during sleeps, assess cognitive functions, and even enable brain-computer interface technologies. Overall, EEG is a powerful tool that allows researchers, physicians, and scientists to gain insights into the electrical pattern and dynamics of the brain, contributing to our understanding of how brain works and how it can be affected by different conditions. Certainly, electroencephalography plays a crucial role in understanding the brain function and diagnosing neurological disorders. Here's why the EEG is so important in these areas. First is understanding the brain functions by real-time insight. EEG provide a real-time information about the electrical activities of the brain. It allows the researchers and the physicians to observe the brain activities as it happens, offering the insight to how different regions of the brain communicate and work together. In cognitive processing, by analyzing the EEG pattern, researcher can study cognitive processes like attention, memory, and decision making. EEG recording helps us in understanding how the different regions contribute to this process and how they change under various conditions. In state of consciousness, EEG ability to capture different brain waves pattern enable us to explore different state of consciousness such as wakefulness, relaxation, deep sleep, and even meditative states. This helps us to understand the neural mechanism underlying these states. So, EEG is important in diagnosing neurological disorders, epilepsies, diagnosis, and monitoring. EEG is a cornerstone in diagnosing and monitoring epilepsies. It helps identifying abnormal patterns of electrical activities that are characteristic of guiding treatment decision and assessing the effectiveness of interventions. EEG is a vital in diagnosing sleep disorders such as sleep apnea and insomnia. It helps track sleep stage and identify disruption in sleep patterns, aiding in appropriate treatment planning. Neurological conditions, EEG assess in diagnosing various neurological conditions like Alzheimer's diseases, Parkinson's diseases, and traumatic brain injury. Changing in EEG patterns can provide early indications of these disorders, allowing for timely interventions. Brain abnormalities. ECG can reveal the brain abnormalities such as tumor and lesion by detecting unusual electrical pattern. It assists in localizing the organs of abnormal activities and guide medical decisions for further evaluations. Coma assessment. EEG is used to assess the brain activities in patient which altered consciousness such as those in a coma. It helps determining the level of brain functions and predict potential recovery outcomes. In neurodevelopment disorders, in children, EEG aids in diagnosing neurodevelopment disorders like attention deficiencies and hyperactivity disorder. In summary, EEG is a valuable tool for gaining insight into brain function both in normal condition and when disorders are present. Its ability to capture real-time brain activities and detect abnormalities makes it essential diagnostics and research tool, allowing us to delve into the complexities of the brain and improve our understanding of neurological conditions. Now, let us talk about the fundamentals of electroencephalography, explaining the basic principles of EEG. Now, let us discuss how electrodes placed on the scalp detect and record electrical signals generated by the neurons. If we talk of the basic principle of the EEG, it operates on the fundamental principle that the brain activities generate electrical signals due to the communication between neurons. These electrical signals, often referred to as brain waves, are crucial indicator of brain function, cognitive states and neurological disorder. The EEG detects and recording process including the following steps. The first one is electrode placement. EEG begins with the placement of the electrode on the scalp. These electrodes are typically positioned in specific patterns and configurations to capture the electrical activities from different regions of the brain. The second one is electrical potential. Neurons communicate by sending electrical signals in the form of action potentials. When millions of neurons work together, they combine electrical activity, generated measurable potential difference on the scalp. The third one is extracellular current flow. When the neuron fibers ion movement creates brief changes in electrical potentials in the extracellular space, this extracellular current flows leads to voltage fluctuations on the scalp, which can be detected by the electrode. The next step is amplification and recording. The voltage fluctuations are extremely small and required amplifications to be detectable. EEG equipment amplifies this signal to make them suitable for recording. The amplified signals are then recorded as a time series of the voltage change over specific interval. The fifth step is signal processing. The recorded EEG signal is often subjected to various signal processing technique. Filtering is commonly used to remove unwanted noise and focus on a specific frequency band of interest. Additionally, data can be segmented to analyze a specific event or brain status. Finally, the interpretation step. The resulted EEG waveform is presented graphically as a series of wave pattern with different frequencies. Each frequency band is associated with different cognitive and physiological state. Now, the challenges and the considerations we are facing while recording EEGs are scalp conductivity. The conductivity of the scalp, skull or brain tissues influence the propagation of the electrical signal. The skull acts as a barrier, distortion and attenuating the signal. Different tissues having different resistance affecting the recorded signal quality. Volume conduction. Electrical signals generated by the neurons spread across the brain and the volume conduct through the surrounding tissues. As a result, the signal recorded at specific scalp electrode represent the summation of the activities of various brain region. Reference electrode. EEG signals are relative, meaning they are measured with respect to a reference electrode. The choice of the reference electrode can impact the interpretation of the recorded data. The next factor is artifact removal. EEG recording can be affected by various artifacts such as muscles activity, eye movement and electrical interferences. These artifacts need to be identified and removed to extract the true brain activity. In summary, EEG is based on the principle of detecting and recording the electrical signals generated by neurons during their communication. Through the strategic placements of electrode on the scalp and amplification of the signal, EEG provides valuable insights into the brain function, cognitive process and neurological disorder. Through careful considerations of challenges and accurate interpretation is crucial for meaningful analysis. The different brain wave pattern alpha, beta, theta and gamma and their associated status. Certainly, the brain patterns are distinct electrical oscillations that can be observed in electroencephalography recording. Each brain pattern is associated with specific mental status, cognitive process and physiological conditions. Here is an overview of the different wave patterns and their associated status. Alpha waves, its frequency ranges from 8 to 13 cycles per second. Alpha waves are typically observed when a person is awake, but in relaxed status with closed eyes. They are often associated with calm and peaceful mental status. Alpha waves are commonly present during state of meditations, deep relaxations and daydreaming. The second wave is the beta wave. It ranges from 14 to 30 hertz. Beta waves are sent when a person is awake, alert and actively engaged in mental activities. They are linked to focus attention, problem solving and cognitive process. Beta waves are prominent during tasks that required concentration, logical thinking and active decision making. Theta waves. Its range from 4 to 7 hertz. Theta waves are often associated during light sleep, daydreaming and creative thinking. They are associated with dreamy and imaginative mental status. Theta waves can occur during the early stage of sleep, during the state between wakefulness and sleep and in states of deep relaxations. The next wave is the delta wave. Its frequency ranges from 0.5 to 4 hertz. Delta waves are primarily seen during deep sleep stage, include slow wave sleep. They reflect a state of unconsciousness and restorative rest. Delta waves are most prominent during sleep, when the body and mind undergo essential repair and relaxation process. The next wave is the gamma wave. It ranges from 30 to 100 hertz and even higher frequencies. Gamma waves are linked to heightened cognitive processing, attention and complex problem solving. They are thought to reflect synchronous neural activity across the multiple brain region. Gamma waves are associated with the tasks that demand integration of sensory informations and coordination of various brain areas such as precision learning and consciousness awareness. It is important to note that the association between brain wave patterns and specific mental state is not always straightforward. Brain activity is complex and influenced by a various of factors, including the individual variation context, external stimulus. Additionally, brain wave patterns can overlap and change rapidly depend on the ongoing mental process. While the relationship between the brain patterns and mental status is still being explored, under this pattern provides valuable insight into the brain dynamics nature and its response to different cognitive and physiological conditions. The role of the frequency and amplitude in interpreting the ECG data is very important. The frequency and amplitude are the fundamental components of EEG data. They plays a crucial role in interpreting the electrical activities of the brain. They provide the valuable insights into the brain function. Different brain wave patterns are categorized based on their frequency ranges. Each brain wave pattern correspond to specific mental states and cognitive process. As we have already seen, the alpha, beta, theta, delta and gamma waves. Abnormal frequency patterns can indicate neurological disorder. Amplitude is also playing an important role in diagnosing. Amplitude reflects the magnitude of the neural synchronization or desynchronization. Larger amplitude includes more synchronized firing of neurons, while smaller amplitude signifies more desynchronized activities. Higher amplitude in certain frequency band can signify distinct cognitive processes. For instance, during focus attention, beta waves amplitude tend to be higher. Now, let us discuss the various applications of the EEG. Certainly, the EEG has a wide range of applications in biomedical field. Here are some of the key applications in clinical diagnostics, aiding in determination of EEG is primary tool for diagnosing and monitoring the epilepsies. EEG is an essential for diagnosing various sleep disorders. It tracks sleep stages, breathing patterns and disruption in sleep cycle. EEG is used to monitor the brain functions in patients with traumatic brain injury. EEG is widely employed in cognitive research to study brain responses to various stimulus, tasks and cognitive processes. It provides insight into how brain process information, attention, memory, decision making and emotional reactions. EEG based brain computer interfaces allows individual with severe motor disabilities to communicate and control external devices using their brain signal. This technology translates EEG patterns into commands for computer applications, robotic device and assistive technology. So, these are the various aspects of EEG. With this, we come to an end of today's session. Thank you everyone for being such a wonderful and engaged audience throughout this session on electroencephalography. Your participation and attention has truly made this class a rewarding experience. It is fascinating to see your curiosity and enthusiasm for delving into the world of EEG. As we come to an end of today's lecture, I want to take a moment to express my heartfelt appreciations to each and every one of you. Before we conclude, I want to appreciate your decisions to learn and your enthusiasm for today's topic. Your engagement has made my role as an educator incredibly fulfilling. Remember that learning is a continuous journey. I encourage you to explore EEG further and discover the countless opportunities it holds. With that, I do like to thank you once again for joining me today. Your presence and participation has made this lecture a meaningful experience for all of us. I wish you all the best in your academics pursuit and beyond. Thank you and have a fantastic day.